Hi, everyone. My name is Julia Hajjar. I'm a PhD candidate in the Population Health Program at the University of Ottawa, and I'm going to talk today about the HPA axis in endocrine disrupting chemicals. I'm also going to talk a little bit about my master's thesis, which looked at bisphenol A and its impact on rat pup HPA axis development and morphometric parameters during gestation and lactation, as well as some common endocrine disruptors and how you can reduce your exposure. So we'll get started now, and I hope you find this presentation interesting. So as we've learned in previous modules, endocrine disrupting chemicals are ubiquitous, they're omnipresent in the environment, and they're exogenous substances that can mimic or interfere with the endocrine system. So the official definition from the World Health Organization is that endocrine disruptors are an exogenous substance or mixture that alter function of the endocrine system and consequently causes adverse health effects in an intact organism or its progeny or subpopulations. So if you bought a coffee, a bagel, a donut, or lottery ticket this morning and took your receipt, you've already been exposed to at least one type of endocrine disrupting chemical already. So endocrine disruptors are really everywhere. They're even on thermal paper, so on the paper that's typically on receipts. So that's another one to keep in mind when you're looking to reduce your exposure to endocrine disrupting chemicals in your day-to-day life. So common examples of EDCs that have been addressed in class already include phthalates, BPA, diethylstilbestrol, parabens, DDT, polychlorinated biphenyls, and PFAS chemicals, just to name a few. So endocrine disrupting chemicals matter because they're everywhere. They affect all of us, and they're there in chronic super low doses most of the time with things like BPA and phthalates. And their effects are largely overlooked, and they have potential delayed effects. So effects can occur years after exposure, and adverse health effects may affect multiple generations, which is why it's really important to really focus in on this topic because the impact is huge. So an insult during a sensitive period in utero or early in postnatal development can lead to altered programming of tissue structure and function, which is what we focused on in this class. So this can contribute to the development of behavioral problems later in life, learning difficulties, altered or delayed cognitive development, cognitive decline, neuroendocrine issues, and other diseases. So endocrine disruptors are also contributing to these types of health issues. So it's really important to learn about them a little bit more. So endocrine disruptors affect us because they can modulate important biological processes such as organ development, reproduction, immunity, metabolism, and behavior. So in recent years, BPA and phthalates, especially in Canada, have moved to the forefront of research because there's concern regarding their impact during the critical window of pregnancy and fetal development in humans. So increased exposure to phthalates during the early phase of the first trimester of pregnancy, for example, has been linked to higher concentrations of estrogen and lower concentrations of testosterone in the fetus. This can affect normal male development in utero and increases the chances of abnormal genital development in male babies. And female reproductive development is also hugely impacted by endocrine disruptors, as is fertility and onset of menopause. There's also been a decline in male reproductive health in recent years. Sperm count and testosterone levels have been reduced, and semen parameters, which are used to measure sperm quality and can help us predict male infertility, are important markers as well. So poor semen quality has been linked to exposure to certain endocrine disruptors, and this has also been observed in recent years. So some examples of endocrine disruptors are diethylstilbestrol, which has been talked about in class already, but Des daughters exposed in utero were at much higher risk of developing clear cell adenocarcinomas, as well as breast, pancreatic, cervical, and vaginal cancers. This is a very potent xenoestrogen. Phthalates are another example. These make plastic flexible, and they're found in a lot of household and cosmetic products, including nail polish, hairspray, and shampoo. Phytoestrogens are found in our food, so this can include things like soybeans, chickpeas, tofu, edamame, pesticides like DDT, and PFAS chemicals, which are typically found in nonstick cookware. So EDCs we know are omnipresent, they're very prevalent in the environment, and they're persistent. So they really don't break down in the environment. So things like birth control pills or things that are flushed down the toilet and enter our water supply will then leak into our oceans, into our lakes and rivers, and the wildlife will then be exposed and will be exposed, and it's just a continuous cycle. So there's been a lot of evidence emerging against endocrine disruptors. In 2015, the Endocrine Society analyzed over 1,500 studies on EDCs and released a report showing that they disrupt hormone function and provided evidence that they're involved in a myriad of health issues, including osteoporosis, obesity, reproductive disorders, hormone-related cancers, just to name a few. So let's talk a little bit about obesogens. So obesogens are endocrine disruptors that influence obesity. So they affect energy metabolism, adipose tissue structure, lipid and glucose metabolism. They're mainly lipophilic in structure and they bioaccumulate. So this is a destructive cycle because this then promotes the retention of other lipophilic pollutant chemicals. So they do this several ways. They can alter insulin sensitivity. They can alter lipid and glucose metabolism. They can increase the size of adipocytes and the number as well. So these are an important group of endocrine disruptors to keep watch on. So these are just more examples of obesogens. So parabens, BPA, and phthalates, which you might commonly hear about, are also considered to be obesogens. So let's get into the nuclear level. So we'll talk about nuclear hormone receptors. So steroid hormone receptors include the estrogen receptors, androgen receptors, mineralocorticoid receptors, glucocorticoid receptors. So we'll focus on the glucocorticoid receptors for this lecture today. So the hormone receptor complex acts as a transcription factor, and it can augment or suppress transcription in particular genes by its action on DNA. So some endocrine disrupting chemicals can bind directly to these receptors, and then they can act as agonists or antagonists, and then enhance or inhibit the effect. So glucocorticoids, as we know, exert effects throughout the body. They're really important for immune system, bones, muscle cells, fat tissue, brain function. An impaired glucocorticoid activity is associated with birth defects, mood and cognitive disorders, cancer, immune disease, metabolic dysfunction, cardiovascular disease, a whole range of health issues. And they also modulate a bunch of genes, including fatty acid synthase, which is key for metabolism, as well as protein coding nuclear factors, which are implicated in anti-inflammatory activity. So glucocorticoids and glucocorticoid receptor and mineralocorticoid receptor binding. So glucocorticoids bind to intracellular receptors. They can bind to the mineralocorticoid receptors at a high affinity and glucocorticoid receptors at a lower affinity. So they're lipophilic and they bind to receptors in the cytoplasm. Once they're bound, the activated receptor complex undergoes nuclear translocation, travels into the nucleus. Once it gets to the nucleus, the receptor complex then acts as a transcription factor and binds to specific glucocorticoid response elements. And then at this point, transcriptional response to stress and local epigenetic changes occur in a cell type specific manner. So this is just an image of how this works. So the mechanism of action of the glucocorticoid receptor. So it's a member of the nuclear receptor superfamily of ligand-dependent transcription factors. And this is just an image I created to show the process. So how does it work? The glucocorticoid receptor bound to a ligand initiates or represses gene transcription. This can be for thousands of genes. So it can bind directly to DNA response elements and or can physically associate with other transcription factors. So the glucocorticoid receptor does a lot in the body, and it's really important to understand the depth and the importance of this receptor, because when we talk a little bit later about how endocrine disruptors can modulate this glucocorticoid receptor, you'll be able to understand a little bit more how important this impact is of endocrine disruptors on HPA axis function. So estrogens can also modulate the glucocorticoid receptor. So this can occur a few ways. The first is by proteasomal degradation by estrogen ubiquitin ligase of the glucocorticoid receptor. So that's the first pathway. The second pathway is through GR phosphorylation inhibition by estrogenic enzymes. And the third pathway is estrogenic repression of GR expression. So this can actually lead to decreased GR gene expression overall and a decreased number of GR available to bind to different ligands. So this is really important to see how estrogen can really modulate the number and availability and function of the glucocorticoid receptor. So estrogen receptors and glucocorticoid receptor activity. So in both the estrogen receptor and the glucocorticoid receptors are expressed in a certain tissue, glucocorticoids can often drive the opposite actions of estrogen. So this is seen in things like the mammary gland and the bone, just for an example. So in the mammary gland, glucocorticoids can drive anti-proliferative activity, while estrogens can actually promote cell growth and proliferation. And in the bone, glucocorticoids can promote bone resorption, while estrogens will block bone resorption. So these two interact very tightly in hormonal systems throughout the body. So let's get into the HPA axis and negative feedback loops. So the HPA axis, as we've learned before, is composed of the hypothalamus, anterior pituitary, and the adrenal gland. So the way it works is the hypothalamus, at the level of the hypothalamus, corticotropic hormone will be released and will bind to the anterior pituitary. At the anterior pituitary, adrenocorticotropic hormone will be released and then will travel to the adrenal gland where it'll bind to its receptors there. And then this initiates the secretion of cortisol. And then cortisol will exert its effects throughout the body. But the important part here with the negative feedback loop is that it's able to turn off when the body is no longer needing this upregulation of the axis. So it wants to return to homeostasis. So what happens when estrogen is present or endocrine disruptors that act as estrogen is negative regulation is really dysregulated. So it'll downregulate glucocorticoid receptor binding, it'll disrupt negative feedback, and ultimately it will result in the upregulation of the HPA axis. So as we talked about before, estrogen can modulate the glucocorticoid receptor. And this is really important because this will throw off the negative feedback loop of the HPA axis. And as a consequence, this pathway won't turn off and the adrenal gland will continue to secrete cortisol and it won't get the message to turn off. So over time, increased amounts of cortisol circulating can cause acute health problems, and chronic high cortisol levels can cause immune issues later in life, chronic high stress, high blood pressure. So it's really important that this axis is modulated properly. So we've talked about the placenta a lot in the course. We know that it connects the fetus and the uterus. It develops to ensure that the fetus receives oxygen and nutrients. And it's an important barrier to maternal glucocorticoids due to 11-beta HSD. So 11-beta HSD converts cortisol or corticosterone in the rat into the inactive metabolites, which then prevent glucocorticoid receptors from activating and make sure that the fetus isn't exposed to high levels of glucocorticoids while its brain is still developing, while it's still in a critical window of development. So placental hormone secretion and fetal growth occur in tandem, as we've learned. And this is just an image of how the 11-beta HSD enzymes work. They're involved in steroid hormone physiology. So type 1 will convert cortisone to active cortisol, and type 2 will do the opposite. So this is really important in regulating the amount of glucocorticoids that transfer from the placenta to the fetus. So in terms of prenatal stressful insults, there's two major biological mechanisms that are involved in modulating fetal trajectory after prenatal stressful insult. So the first is maternal immune activation, and the second is elevated glucocorticoids, which we'll focus on. So as you know, consequences of elevated glucocorticoids include an upregulation of glucocorticoid signaling to the fetus, which has downstream effects on neurodevelopment. And glucocorticoid exposure during fetal development is a critical mechanism that underlies prenatal programming of adult affective behaviors such as depression and anxiety. So this is a diagram that I made just to demonstrate placental estrogen synthesis. So first, the maternal HPA axis releases DHEAS, which travels to the placenta and then into the fetal bloodstream where it activates the fetal pituitary. This results in the release of DHEAS and glucocorticoids. Fetal DHEAS will then provide the placenta with the substrate it needs to synthesize estrogen, which is important because the placenta lacks its own DHEAS. In instances of a high estrogen pregnancy, fetal ACTH is reduced, resulting in a lowered production of DHEAS by the fetal adrenal. And because of this lowered production of DHEAS at the level of the fetus, overall estrogen production will then be lowered in the placenta. So it's really important to see this dynamic and how important secretion at the level of the fetus is to regulate DHEAS in the maternal supply. So in terms of EDCs and pregnancy, exposure during pregnancy has the most serious consequences because this is such a critical window of fetal development. So disruptions during this time period can cause permanent effects due to the brain still developing, this being a really critical window of neurodevelopment. So EDCs have been widely detected globally in maternal urine, blood, serum, amniotic fluid, breast milk. So steroid hormones are lipophilic and they can accumulate in our fatty tissues. They can transfer from the mother to the fetus through the placenta, and they can transfer from the mother to the infant through breast milk. So what the mom is drinking or eating is really important because that'll transfer directly to the fetus. So ironically, in BPA research and endocrine disruptor research, it's during this most critical time in development that there remains a significant gap in the literature. So we know that EDCs with emerging literature have shown, that EDCs have caused permanent developmental consequences and damages to physiology and consequences to behavior if exposure has occurred during this plastic period. So things like exploratory behaviors have been altered because of exposure to things like BPA and phthalates during development. So we'll talk a little bit more about BPA here because my thesis was focused on BPA. So from the previous lecture, we know that it's an organic compound. It's used to synthesize epoxy resins, and it's found in a lot of plastic products, including water bottles, plastic toys, can linings, medical devices, and even thermal paper receipts like I talked about before. So 15 billion pounds of BPA is produced roughly per year. And the predominant routes of exposure are oral ingestion, absorption through the skin, and inhalation. So one thing to keep in mind with BPA is that it leaches out of plastic. So the polymer breaks down through exposure to high heat or abrasion or acidic substances. So one thing you don't want to do is pour a boiling hot spaghetti sauce into a plastic container because that will help promote the leaching of this BPA, or putting a plastic Tupperware in the dishwasher or in the microwave. So because BPA can mimic estrogen, it can mediate the glucocorticoid receptor and can ultimately decrease glucocorticoid receptor activity and number. So we talked about this before with the interaction between estrogen receptors and estrogenic chemicals and the HPA axis. So we're looking at this in the context of BPA and the glucocorticoid receptors. So BPA can also bind to estrogen receptors alpha and beta, as well as the glucocorticoid receptor. So it's hypothesized to exert glucocorticoid-like action as well. And in Canadians, BPA is quite prevalent. The highest concentrations of BPA in children 3 to 5 and 6 to 11 years of age. So those are really critical windows of development as well for the brain. And infants have the highest food intake and body weight ratio and highest BPA exposure levels due to this food intake and body weight ratio. And unfortunately, this is also a very critical window of development. So exposure is quite high in this group too. So it's detected, as I mentioned before, in the urine of over 95% of Canadians, as well as in breast milk, cord blood, and even meconium. So health consequences of BPA include increased rates of prostate and breast cancer, decreased fertility, reduced semen quality, urogenital abnormalities in male babies, earlier puberty in girls, increased body fat and obesity, type 2 diabetes, and neurobehavioral problems like ADHD. So this is an important chemical to really study because of all of the health consequences that it has. So now we'll talk about the stress hyperresponsive period. So this is a really unique physiological phenomenon in the rat pup, which is used to protect the still developing brain by blunting the HPA axis. So it does this for a period of time between postnatal day 5 and postnatal day 15. So at this time, the HPA axis still works, but it's very, very reduced in activity. So at the level of the adrenal gland, we'll see low corticosterone circulating and a lower affinity for ACTH. So if we have a lower affinity for ACTH at the level of the adrenal gland, it's going to not promote as much secretion of corticosterone. As a result, CRF at the level of the hypothalamus is inhibited and the whole pathway is blunted. So unless it's a life or death situation, the HPA axis is pretty unreactive during the stress hyporesponsive period. So in terms of my thesis, the research question I had and my objectives were pretty interesting. So my research question was, I wanted to see if gestational and lactational exposure to BPA perturbed perinatal development in the rat pup. So my objectives were threefold. The first was to carry out a BPA dose series exposure study in timed pregnant Long Evans rats. The second was to assess gross morphology parameters in the BPA exposed rat pups at 2 time points post-birth, postnatal day 5 and postnatal day 15, which correspond to the beginning and the end of the stress hyperresponsive period that I just talked about. So common morphological parameters that are used in toxicology research are anogenital distance, crown rump length, and body weight. So those are the 3 parameters that I looked at in my study. And the third objective was to characterize the molecular effects of BPA on glucocorticoid receptor gene expression levels in male adrenal glands. So this kind of corresponds to the estrogenic impact of BPA on a component of the HPA axis pathway, like we just talked about. So the tissues that I collected included all of the components of the HPA axis, so the hypothalamus, the anterior pituitary, the adrenal glands, as well as many other tissues that we saved for future projects. So for morphometric measurements, like I mentioned, I looked at body weight at both time points, crown rump length at both time points, and anogenital distance at both time points. So these are just some graphs from my study. So what I found was that in the younger animals, so postnatal day 5, the treatment significantly affected body weight. So the BPA 50 group, so we used ultra low doses of BPA for the project, BPA 5, BPA 50, and BPA 500. So the BPA 50 group we found had significantly heavier animals compared to the vehicle group. So our vehicle group was just a control solution of sucrose, and the BPA groups I dosed with these ultra low doses of BPA. My control group was, or my positive control was diethylstilbestrol, which is a very potent xenoestrogen. So we found there was no significant effect of treatment or sex on body weight observed in the postnatal day 15 animals. So for crown rump length, we found that treatment significantly affected crown rump length. So the DES, or the positive control group, the diethylstilbestrol group, and the BPA 50 groups had significantly longer animals compared to the vehicle group, or the sucrose solution. So we found that sex did not significantly affect crown rump length. And then in postnatal day 15 animals, we found a significant effect of treatment on crown rump length as well. And in terms of anogenital distance, for both postnatal day 5 and 15, male anogenital distances were significantly longer than females at both time points. So this is physiologically expected because male anogenital distances are typically twice the length of female anogenital distances. So we weren't really expecting to see any differences here. Now getting into our molecular measurements, so our glucocorticoid receptor gene expression levels. So we had two reference genes or housekeeping genes. So these genes are used because they're typically not affected by your treatments. So GAPDH and 18S rRNA are components in cells that are present in pretty much all cells, and we weren't expecting BPA to impact these at all. But we ended up finding, which I'll talk about later, is that our 18S rRNA gene was actually impacted by our BPA exposure. So this became a target gene. So 18S rRNA is present in all eukaryotic cells, and it's structural RNA for cytoplasmic ribosomes. GAPDH is an enzyme of the glycolytic pathway, and high levels are present in all cells. So it's well established in the literature that these will not be affected by circulating estradiol or BPA, so which makes them really good housekeeping genes or validation genes. So in terms of our molecular results, we found that our fold changes, which represented GR gene expression levels, downregulation, so a decrease in GR gene expression, was consistently observed in our positive control group, so the diethylstilbestrol group, in both age categories. So both at the young age category of postnatal day 5 and at postnatal day 15. And we also saw upregulated fold changes observed in the postnatal day 5 BPA 50 pups, which means GR gene expression was increased, and at the postnatal day 15 BPA 500 pups. So this was biologically relevant. So when we're looking at our reference gene, which then became our target gene, 18S rRNA, we saw downregulation of our fold changes or a lower GR gene expression in our postnatal day 5 diethylstilbestrol group, our BPA 5 and our BPA 500 group. So this was something we were not anticipating to happen because this is supposed to be a housekeeping gene. We didn't expect to see any effects. And in the postnatal day 5 BPA 50 group, we saw an upregulation, so an upregulated GR gene expression. And then we didn't see any changes in the postnatal day 15 animals. So this was something that was sensitive for the younger age category. So the animals right before they entered the stress hyperresponsive period. So the conclusions for my study for the morphometric parameters, so crown rump length, anogenital distance, and body weight, was that the BPA 50 treatment group had significantly increased body weights and crown rump lengths, but only at postnatal day 5 at the younger age group. So this might suggest a developmental sensitivity to BPA, which is apparent at postnatal day 5, but not at postnatal day 15. And in terms of the molecular parameters, the diethylstilbestrol treatment group demonstrated consistent downregulation in male adrenal gland GR gene expression at both postnatal day 5 and postnatal day 15. So BPA sensitivity we also found was developmentally stage specific. So it was affected at postnatal day 5, but not always at postnatal day 15. And the DES treatment group, because it's such a potent xenoestrogen, it showed that it did downregulate GR gene expression, which was interesting. And the unanticipated portion of our findings was that this 18S rRNA housekeeping gene was actually sensitive to BPA in the younger animals in postnatal day 5. So dose response curves are interesting because this is actually what we found in my study, was that we seem to see a non-monotonic dose response curve in some of our age categories and monotonic in others. But with BPA generally, non-monotonic dose response curves are what you'll typically see. And this is interesting because EDCs are regularly linked to these non-monotonic dose response curves, but policy has been formed using monotonic curves. So a lot of policy needs to be updated to reflect this. And it's hard to kind of get past the concept of the dose makes the poison, which is typically seen with monotonic dose response curves. So this is a really interesting area of study as well. And this is just one final slide of fun facts. So did you know that there are controversies in the field of BPA research? So three big ones are the effects of these toxicants at low doses. So this low dose controversy has been quite prevalent with BPA research and other EDCs, but more studies are emerging showing that there are significant health impacts and effects at super low doses of endocrine disruptors. The second is that the source of funding may influence study outcomes, which was actually shown with BPA. And BPA was actually the driver of requiring conflict of interest statements in research articles. So findings in some industry-funded studies for BPA consistently showed no adverse effects due to low dose BPA exposure, while hundreds of studies, industry or private studies or university studies showed that there were adverse effects at super low doses. So they ignored these over 150 university studies in favor of 11 industry-funded studies that showed no adverse effects. And that's why a lot of these BPA regulations were formulated. So that's really interesting. And the third is the monotonicity, which I discussed earlier. And it's also important to be mindful of greenwashing. So consumers are often unaware that BPA-free doesn't necessarily mean estrogen-free. So most products labeled BPA-free still have estrogenic activity, and oftentimes the A is replaced with another equally estrogenic chemical. So plastics labeled BPA-free in one study by Yang actually had the highest amounts of estrogen leaching. So I thought that was really interesting. So your best bet is to opt for a stainless steel or glass or silicone and kind of shift away from BPA-free labeling. So I hope this presentation was interesting for you, and I hope you learned something new, and please let me know if you have any questions.